Three main activities for the nervous system are sensory functions, integrating functions, and motor functions. So the nervous system senses changes from within the body or from outside the body and conveys this information to the spinal cord and brain. The spinal cord and brain then senses the information that is received, so the sensory function, and it is analyzed, stored, and integrated to produce a response. So that's the integration. And the motor response instructs the body to do something, such as contract a muscle or cause a gland to secrete its products. Neurons or nerve cells are the basic functional units of the nervous system. They cannot reproduce, but they can regenerate. So shortly after an animal is born, its neurons lose their ability to reproduce, but they can regenerate cell processes if the body remains intact. So the lack of reproductive ability is why serious nervous system injuries, such as strokes and spinal cord damage, are often so debilitating and have long-lasting effects. They have a high requirement for oxygen, and this is why even after minutes of lack of oxygen, they can start to die, and this is why CPR is so important to be started after an arrest. First, doing the chest compressions, you're moving oxygen around, that the blood that's already oxygenated, and then you need to start supplying oxygen. And they have a support and protection from neuroglial cells. So the neuroglial cells are structurally and functionally support and protect the neurons. So they are important parts of the infrastructure necessary for the neurons to be able to do their job. Dendrite cell processes. The dendrites are the projections at the end of the cell body, and they are responsible for receiving stimuli from other neurons, and they conduct the stimuli toward the cell body. So afferent, again, it starts with an A, so it's kind of like adding. Efferent is away, so there's no good way to remember that. But afferent, just think of adding. They also serve as sensory receptors, so for heat, cold, touch, pressure, and stretch. The axon of the neuron is kind of like the tail. So it is a long process that may be covered in myelin, and it conducts nerve impulses away from the cell body. So it is an efferent process, so away toward another neuron or effector cell, such as nerve fibers or bundles of muscles or gland cells. For myelinated axons, it is mostly just important to remember that myelinated axons conduct impulses faster than those without a myelin sheath. So the myelin sheath is made of cell membranes of specialized glial cells called oligodendrocytes in the brain and spinal cord, and Schwann cells in the nerves outside of the brain and spinal cord. And basically they are wrapped around the axon, which makes the impulses go faster. And in between the glial cells are small gaps in the myelin sheath called nodes of Ranvier. And these work together to enhance the speed of conduction of the nerve impulse along the axon. So those are the main points to remember when asked about myelinated versus non-myelinated. The two main systems of the nervous system are the central nervous system, or the CNS, or the peripheral nervous system, or the PNS. And basically, it is right in the name where they are coming from. So the central nervous system is composed of brain and spinal cord and found along the central axis of the body, so central nervous system. And peripheral is the components that extend away from the axis in the center, so peripheral, and they are the cranial nerves and the spinal nerves. I think this is a difficult concept to grasp, but in general, if you are to think of polarization being negative, then that might help. So forget the words de and repolarization and just think of the word polarization as being negative. So the resting state of a cell is negative, and to depolarize that, or to effectively make it not negative anymore. This is when the sodium channel um, is open and the potassium from outside the cell influxes into the cell, making it go from a negative state to a positive state. And the repolarization, or making that cell potential go from its negative state to a positive and then back to negative again, or repolarizing. This is done from the potassium from inside of the cell moving outside of the cell. So polarization equal negative, depolarization is going from negative to positive, and repolarization is going from positive back to negative, which is where they start from. So this slide and graph are showing the repolarization, or how the cell is going from more positive to more negative, and it is the opposite of depolarization, but they should put that the sodium ions have two pluses on them when they do these graphs, because I think that would help. So I've added two pluses to the word Na in this slide. So if you were to think of the graph being the opposite from what it's showing right now, but that two sodium ions that have two pluses on it versus the potassium only having one plus, and three of those ions going from the outside to the inside, then obviously those sodium ions that have more pluses on them are making the inside of that cell more positive. And in the repolarization, which is shown here, this shows that those more positively charged sodium ions are now moving out of the cell, and the less charged potassium ions are moving back inside the cell. So it's going from having way more sodium ions inside the cell that have two pluses on them versus the one plus for the potassium. So that would make it more positive. And when they leave in the repolarization state shown here, they are moving the more positively charged sodium ions out of the cell, and some of the less positively charged potassium ions are going back in. So now the outside of the cell is more positively charged than the inside. The refractory period is a very brief time period when a neuron is insensitive to additional stimuli. So this depolarization and repolarization of a cell is literally happening in seconds. But right in between the depolarization and repolarization, there is a time period where the neuron is not able to take on any more work because it is busy with the depolarization and repolarization. So that is what the refractory period is. Just think of it as the neuron is busy trying to do all this work and it just can't take on any more work. So this is basically showing a synaptic transmission where the impulse starts at the neuron and it goes down the axon to the telodendron to the synaptic knob, and then the vesicles end up releasing their neurotransmitters through the end of the knob into the synaptic cleft, so the area that is between the synaptic knob and the next receptor. And this is where the neurotransmitters are released into so they can be taken up by the next cell by the receptor molecules that are embedded in that cell. In synaptic transmission, the nerve impulse from one neuron to the next cell travels down the axon to the telodendron to the synaptic knob, which is pictured here. And the depolarization waves cause the vesicles within that synaptic knob to dump the neurotransmitter into the synaptic cleft, which is that area or space in between the end of the synaptic knob of one neuron to the receptor end of another cell. And at the receptor end, there are receptor molecules embedded in that that are able to take on that neurotransmitter. The excitatory versus inhibitory neurotransmitters, I guess if you can just think of the excitatory ones being really excited for the influx of the positive sodium ions, so that moves their threshold closer and they're able to accept these positive ions. The inhibitory ones are really negative, and your textbook describes them as being hyperpolarized, so hyper negative, and these ones are moving that threshold farther away. So excitatory is really excited for the positive influx of sodium, and inhibitory is really negative. The neurotransmitters that we mostly deal with are acetylcholine. So the acetylcholine, where the sympathetic nervous system takes part in, would have an inhibitory effect, which would slow the heart rate. The catecholamines are norepinephrine, epinephrine, and dopamine. And if you think of these in terms of shock related to anaphylactic allergic reaction, then we use epinephrine and norepinephrine in those situations, and that would kind of have an excitatory effect or wake up their sympathetic nervous system for the fight or flight response. And a lot of these patients have low blood pressure, and the same drugs are given in anesthesia if they're having low blood pressure. So norepinephrine and dopamine are commonly used. So that again would be an excitatory response to the sympathetic nervous system to increase the blood pressure. GABA aminobutyric acid, or GABA, that is an area in the brain that a lot of these work on. So it is an inhibitory response or an inhibitory neurotransmitter. And if you think of Valium having an increased effect on the GABA, which is an inhibitory activity, that would produce tranquilization. So if you think of GABA being a really sedate kind of state, then if you're increasing the GABA, you're increasing the drowsiness or tranquilization. The cerebrum is the largest part of the brain in domestic animals and is associated with learning and intelligence. The surface is covered with gyri, separated by fissures, and sulci. So the gyri are the hills, and the sulci are the basically depressions. And longitudinal fissure divided into right and left hemispheres goes right down the middle. Each hemisphere of the cerebrum is divided by sulci into lobes, and they all have specialized or different functions. So there's the frontal lobe, parietal, occipital, temporal, pons, medulla, and the cerebellum at the back there. The cerebellum is the second largest component of the brain, and it is located caudal to the cerebrum. It is the area responsible for coordinated movement, balance, posture, and complex reflexes. Damage or disease to this area of the brain results in hypermetria or really exaggerated gait. So a lot of these animals that have damage or are born with insufficient amount or an area of the cerebellum that is not working properly have something called cerebellar hypoplasia, and they have this really hypermetric gait where they can't really coordinate, or it's like they don't know what their legs are doing, and they have a really exaggerated movement when they are walking. So the brain stem is the connection between the spinal cord and the rest of the brain. There are different components. So there's the diencephalon, which has the thalamus and hypothalamus. So di meaning two, so there's only two components there. The brain stem has three, so it could be called the triencephalon, but they don't. So brainstem has the midbrain, the pons, and the medulla. And the brainstem is responsible for maintaining basic support functions of the body. So without your brainstem, you would not be able to live, because you would not be able to breathe, have your heart function, and other things such as vasomotor control. So the ability to control the diameter of your blood vessels, also controls swallowing. So brainstem is something that operates at the subconscious level, and you can't live without it if it's damaged. Cerebrospinal fluid, or CSF, is a fluid that is between the layer of the meninges and in canals and cavities inside the brain and spinal cord. So this provides cushioning, may play a role in the regulation of autonomic functions such as respiration and vomiting. And CSF taps are done to be able to have a test sample for diagnosis in certain diseases or cancers. This is something that we do quite often because we have a neurologist. So they will tap the spinal fluid either usually right at the base of the head, so at the back of the atlanto-occipital area, and you can see that it is cloudy. It may contain too much protein or not enough protein, bacteria, white blood cells, all of these that are able to give us some insight to why an animal may have um, like seizures or other diseases that affect the brain. The blood-brain barrier is something that will come up when dealing with certain drugs. So if you're wanting to target the brain itself, then you need to choose drugs that are able to cross across this blood-brain barrier. So it is capillary walls in the brain that have no fenestrations, covered by cell membranes of glial cells, and it is basically a layer of protection that prevents proteins, ions, drugs, and other molecules from readily passing from the blood into the brain. The autonomic nervous system is split into two different systems, so the sympathetic nervous system and the parasympathetic. Sympathetic being the nerves that are emerging from the thoracic and lumbar vertebrae, so the thoracolumbar system, and the parasympathetic nervous system emerging from the brain and the sacral vertebral regions. Here is that word once again, efferent, meaning away from. So these are the nerve impulses that are traveling from the central nervous system away from it to the preganglionic neuron and postganglionic neuron. As pictured 2 slides ago, the sympathetic nervous system has preganglionic neurons that either may synapse with a neuron within a ganglion chain or pass through a ganglionic chain and synapse with a neuron located beyond the chain. So that's basically those chains coming off of the CNS and then branching into several other chains and then maybe meeting its target organ after. And the opposite is true for the parasympathetic system. So as the slide showed now 3 slides ago, the parasympathetic system has a much shorter process to reach its target organ. So the parasympathetic preganglionic neuron travels directly from the CNS to its target organ. The synapse within a short postganglionic neuron in the target organ. So it has short chains that go from the CNS to the postganglionic neuron right to the target organ, rather than the many chains that the sympathetic system can have. So the autonomic nervous system with the sympathetic and parasympathetic, basically you can think of them as the sympathetic system being fight or flight, and because of this, it helps the body cope in emergency situations. And the parasympathetic is rest and restore. It brings the body back to a resting state and facilitates all body processes to replace resources used during the emergency. So these are things like being able to process food in the gastrointestinal tract, which you definitely wouldn't need to be doing if you're in a fight or flight situation. So what is happening with a reflex arc is that a sensory receptor detects a change. The receptor then sends an action potential along sensory neurons to gray matter of the spinal cord or brainstem. The sensory neuron synapses with other neurons for that incoming impulse to be integrated with other sensory neurons, and the integrated response is sent back out by motor neurons to the target organ. So the CNS moderation of reflexes. So with injury, intact reflex arcs caudal to the spinal cord trauma become hyperreflexive. So these are exaggerated reflexes. So hyper meaning too much. With injury where the reflex arc enters or leaves the spinal cord or damage to sensory motor neurons of the reflex arc, then it will be hyporeflexive or absent. So hypo meaning not enough. The stretch reflex is involving the sensory neuron and a motor neuron, and it only has one synapse and no interneurons. An example of a stretch reflex would be the patellar ligament when that is tapped and the force against the ligament is slightly stretched. So the quadriceps and femoris muscles on your thigh are stimulated and result in a contraction of the quadricep. So that is the slight kicking because of the stretching of the ligament. The withdrawal reflex is something that is commonly used to assess spinal cord trauma. So it is where there is a receptor picks up a stimulus, so that would be pinching of a toe really hard, and the effector muscles being able to flex those muscles, producing the withdrawal effect. So if they cannot withdraw their leg from the pinching of the toe or the stimulus, then they may have some spinal cord trauma. So the other clinically significant reflexes, one is the palpebral reflex, and that is when you tap the medial canthus of the eye and it should produce a blink. We use this in anesthesia to assess the depth of anesthetic plane they are in. So if they can still blink, they are not in a appropriate anesthetic plane for surgery. And the other one is the pupillary light reflex, and this is the normal response to shining light into the eye. So the pupil should constrict. So there's either direct or consensual, and direct should be shining a light directly into, say, the right eye. So the direct is the right eye pupil constricting, and consensual is when you shine a light into the right eye, the left eye pupil should also constrict. So that is consensual.